Okay, hello. This is our final exam, part one, Q&A session. So, any questions, go ahead and send them my way. Looks like I'm talking to myself right now. Okay, so while I wait for people to come online, um, let me just remind you of a couple things. Your final exam will cover all of, this portion will cover all of semester one. It will, so I think that's up to like page 82 in your notes. Essentially, the last topic is PCR. And 45 to 50 questions. I'm still finishing that up. And you'll have all multiple choice. It will be scenario based, much like the last one, except that there are no lab based questions. So, no all some or none tables, no plasmid mapping, that kind of thing. Let's see. You can look, I would reference your old final exam that I gave back to you to help you study. I would also recommend that you use the final exam resources from last semester. Hopefully all of those things will help. There's also like the practice finals from last semester are all online and such. So definitely you could watch those. Let me see, what else can I remind you of? I would say it's a pretty broad range test, like you want to know all the stuff from last semester, but don't over focus on teeny tiny things. Like I don't need you to memorize the functional groups again or things like that. What else? Let me see. Of course, if you have any questions, go ahead and type them and I should be able to see them. Let's see. The information you need is all on the website. Yes, phosphorylation. I assume you mean like in terms of enzyme regulation. So the idea with phosphorylation, I mean, at the core, it's the idea that you add a phosphate group to a protein or an enzyme, right? Because remember, enzymes are proteins. And at the core of it is just the idea that if you add something to a protein, you change its shape. You change its shape, you change its function, right? So in this case specifically, what we're adding is a phosphate group. And the idea then is phosphorylation can either activate a protein, an enzyme, or inactivate it, right? Because the idea is that in adding a phosphate, you now change the shape of that molecule, that protein. And when you change its shape, you potentially change its function. So for scenario one is that you have a protein and you phosphorylate, and it's already in its functional form, and you phosphorylate it, and then doing that, you change its shape, and now it no longer works. So you could inactivate a protein that way. Or on the other hand, what could happen is you could have a protein that's in an inactive form. By adding a phosphate, you now change its shape, and that becomes the active form, and now it starts working. So either way, though, it's a way that you can enzymes and proteins can be regulated. It's actually, you kind of actually see it in cellular respiration, which we covered second semester, of course, but it's a good example actually where you see in glycolysis a lot of phosphorylation causing the next intermediate to form. Let's see, what else? Let me see, time range. Did I get you? Yeah. Okay. No problem. Any other questions you guys have? I know it looks like there's not too many people watching right now, so don't know if there's going to be a ton of questions. Let me think. You definitely need to make sure you can identify different biomolecules, you understand how they work, be able to transcribe and translate a protein or DNA into RNA into protein. What else? I think one way of thinking about it is, last semester you had like 90 questions in the final, and this time it will be shorter because it's a shorter period. So on some level, you still have to know the topics, and they're quite wide ranging, but there'll probably be a little less super specific because of that, because there's so many less questions. Let's see. You want to be able to do Punnett squares, understand genetics, understand things like PCR, how they work. Oh, and like mitosis, meiosis, all that stuff. Yes, I can totally go over telomeres and telomerase. So the idea is that when you do DNA replication, just because of the way the enzymes work, you actually can't copy the very end of it. And so inevitably, every time DNA replication happens, the DNA is going to get a tiny bit shorter. And that would be problematic if you were now losing important parts of your genes, right? So instead, what you have is those ends of the chromosome, which are telomeres, which essentially, because of this glitch, I guess you could say, in DNA replication in the process, is going to lose a little bit of that end every time. So the telomeres then are like these repeating ends of the chromosomes that sort of serve as sort of a buffer, so that if, or when, not if, because it's going to happen, when the DNA gets shorter just due to the process, what they're losing is actually junk, right? Not something that's important. That being said, though, since cells divide, and basically when they divide to a certain amount, and then they basically are outdone and they self-destruct. What's going to happen then is that would be a problem, right? If you think about your reproductive cells, because that would mean like if cells in the testes or in the ovaries are dividing, and they are dividing a certain number of times, let's say, right? Then you're going to end up with a situation where they're now going to have telomeres that are shorter. And so if that particular, say, sperm is used to fertilize an egg, it has telomeres that are the length of, maybe it's lost a certain number because of the divisions. So telomerase is an enzyme that, or there's a gene for telomerase in your cells, and it is switched on, is active in cells that are going to become your gametes. What they've also found, though, is that cancer cells are really unique, right? Because you would think that if they were dividing, and after a certain number of divisions, cells essentially self-destruct, you'd think that cancer would destroy itself, and yet it doesn't. So that's because when you have these cancer cells, yes, they're short in their telomeres because they're dividing a whole lot, but they're also actually switching on telomerase, so they add enough back of their telomeres to sort of stay afloat, and that's why cancer cells, seem quote, immortal. Hopefully that helps. Let's see. What other questions? Go ahead and send those my way. I'm gonna pull up, I guess, the practice exam and see if there's anything in there that I noticed you guys need to make sure you know. Keep in mind the practice exam, as it's noted, is not multiple choice, but you should be able to then convert things like this to multiple choice questions. Let's see. Let me just take a look at the practice exam. Again, kind of just see if I notice anything that you need to be aware of. Levels of protein structure you should know, yeah. Endothermic versus exothermic reactions, enzymes and how they're affected, such. So again, if you have any questions, feel free to ask them. I guess it might be good for me to just talk about, evolution. Evolution is semester 2, so you don't need to know evolution for semester 1. Semester 1 is going to start with ecology, and from ecology you go into biomolecules and biochemistry, and then from there you would go to protein synthesis. From protein synthesis you would go to cell signaling, mitosis, meiosis, genetics. Yeah, that's everything. Again, essentially from pages 1 to 1 to 82, I think. Yes, and you can come talk to me later about chi-squared and evolution and Heidelberg, but for now, don't worry about that. Not on the final on Friday. Let's see. Trying to think what else I could remind you of. Oh, that's what I was about to do. So on the practice exam, there is the PCR and designing primers and such, right? Remember the idea is like if you want to copy the DNA, you have to make a little primer that gives it something to bind to, and it would copy what's between those sections, right? So conceptually, make sure you understand that idea too. Well, hi, Matthew. Matthew, just so you know, this is for AP Bio, so you're welcome to listen, but it might not be as relevant to you. For you guys, it's going to be your tomorrow night. We'll do a quiz for you guys. Let's see. What else can I talk to you guys about? AP Bio-wise. Remember, you do need to know the stuff in your notes that has to do with, so it's in your notes, it's fair game, right? So you should know conceptually how something like gel electrophoresis works, or, and restriction enzymes. But again, I won't ask you lab-based questions. That's sort of the distinction. Okay. Let's see. Any other questions? A few of you who are online. Make sure you know your different enzymes. Make sure you don't get confused between like DNA polymerase, RNA polymerase, all those enzymes. Let's see. Oh, I see a question. Okay. Yes, the difference between DNA polymerase 1 and polymerase 3, they're both involved in DNA replication, and they both essentially build new strands of DNA, right? The main building enzyme in DNA replication is going to be DNA polymerase 3. Its job is to take that primer, which was attached using primase, and start to build the new DNA in a fashion of from 5' to 3', or from 5 to 3, right? Whereas, remember, that means that now what you have when you finish that portion of DNA replication is you have a section of DNA, but it's attached to your primer, which is RNA. So what DNA polymerase 1 will actually do is it will cut out the RNA and then help replace it with DNA. So they're both building in DNA, but one is the main builder, which is DNA polymerase 3. And then the polymerase 1 is really kind of just supposed to be functioning off of the, the repair aspect, or how it has to get rid of something first, the RNA primer. Yes, signal transduction, sure. So with signal transduction, remember the overall idea is it's a way that a cell is going to get a signal, receive a signal, and then have some sort of end result. So there are three steps to signal transduction, right? There's reception, transduction, and response. Reception is the idea that some sort of molecule binds to a receptor on the outside of the cell, and that's now going to trigger a whole chain reaction of stuff to happen. That sort of in between steps of all those chain reactions of things that happen, that would be transduction. And then ultimately you end with like some final response, right? And so that's the sort of last portion. No, I don't need you to know the history of inventions and DNA historical scientists. That's not important at all. Let's see, what else? I guess I'll look back at the practice exam, see if you guys have any questions about that. Again, make sure you understand how restriction enzymes, how DNA, how all those things work, because I'm going to, you expect that you understand the concept behind them. Know the processes, know transcription and translation, and all that good stuff. I guess with signal transduction, we can talk about things like amplifying the signal also. The idea that you can like have one molecule trigger 2 and 2 triggers 4 and 4 triggers 8, etc. No, you do not need to know lab stuff. I mean, again, you need to know it in the context of it. If it's on your notes from pages 1 to 82, you're responsible for, right? Like understand conceptually how gel electrophoresis works, but not sitting there and like trying to troubleshoot whether or not they did what you want them to do. Let's see what else. Let me just take a little look. Yeah, so again, the practice final has like the all summer none table practice, and you can just ignore that and don't worry about it. Any questions? And also, can I remind you? It's not really related to the final, but a question I have been getting lately is about the AP exam, or not AP exam, SAT2s, on whether or not you would take the E version or the M version. E stands for ecology, M stands for molecular. As I told, yeah. I'm not sure what I was about to say. Let's see. Other questions? Let's see, what else? Operons. Yes, I can totally go over operons. So the idea behind an operon is that it's a section of related genes, right? I mean, essentially sort of like putting all your stuff from history into like one divider so you can find it later. But in the case of the bacteria, who are the ones who have operons, it's just a way for them to sort of transcribe and translate everything at once. So, for example, you like the lac operon, the trip operon, where each group of enzymes is responsible for doing something. And that way you only transcribe all those genes when you need them, but you transcribe them all at once. A promoter then is just a segment of the DNA that contains a specific size, specific sequence. And that being said, then you'll be able to possibly, bind to and start transcription in finding that promoter. You should know the different types of operons. We have our lac operon and our trip operon, right? Those are repressible and inducible. Trip repressor is operon is a repressible operon, where the idea is that you have these enzymes and proteins that make tryptophan, and so if they, if you need basically, if you need that particular compound, then you can switch on that operon. And then on the other hand, so inducible and repressible, right? Repressible is an operon that's normally on, but you can switch it off. Inducible is an operon that's normally off, but you can switch it on. And I believe we have like animations and videos help you get all that stuff as well. Let's see, what else? The promoters, remember, don't only apply to operons. They are also applying to just any gene, right? It's a certain sequence then, and remember, technically it's 2 binding sites. We would give you those sequences that you look for as an operon. But the idea is that you have to have those 2 slots correctly spaced together, and everything else kind of has to be, lined up. So a promoter is sort of like the start sign for transcription in a cell. What else would you like to ask about? Let's see. Definitely do the practice exam. I think I may have already said that. Yeah, it's kind of. I mean, yeah, the question is the trip operon kind of an example of a negative feedback loop? I mean, to some extent, yes. I mean, because when there's enough tryptophan, it would go back to some of the beginning steps to creating tryptophan, right? And in doing so, then it shuts down temporarily until it needs it again. Sorry, I'm just readjusting for a second. No problem. Sorry, I'm not sure how useful this is because there's not that many questions, but whenever you do questions, I'm happy to help. Let me see if I look at the practice exam. What else do I notice? Where is the activation? Okay, so activation energy is basically like the top of the hill, right? So essentially, the activation energy goes from the energy in the reactants all the way to the top of the hill for like any given situation. That, keep in mind, is going to be a little different depending on if you're talking about endergonic or exergonic reaction, right? Because an endergonic reaction will mean you're putting energy in, exergon taking it out. So don't get caught up in those distinctions. You want to just make sure you can answer the questions. How many questions are there? There are about 45, 47 questions. Like I said, I'm still adjusting it, but that's plenty at the moment. Let's see. I'm not sure what else to talk about. Definitely no endergonic, exergonic. Yes, CAP and CAMP have to do with positive gene regulation in operons. Again, only operons, not humans. Although CAMP is found in a lot of other reactions. So the idea is that you can think of the lac operon, which is our inducible operon, as having two levels of control, right? There's whether or not it's even going to transcribe and translate that operon, but then there's the how fast. So if you have a lac operon and you want to transcribe it, then CAP and CAMP can help. And essentially, CAP is this protein, and when CAMP binds to it, it helps recruit DNA polymerase to that area during transcription. So without the main molecule is CAP. And then CAP can be activated, like CAMP, basically. Or they work together. The lytic cycle, no, does not take place in human cells. It only happens in bacteria. Let's see. Oh, the lytic cycle is the bacteriophage cycle. So it's the cycle where you have bacteriophages, which are viruses that can infect bacteria, right? And they can do one of two things. They can go in, basically make a ton of copies of themselves, burst out, and kill the cell. That's the lytic cycle. Or there's a lysogenic cycle, which is where they go into the cell and they kind of stay dormant, almost like hiding. And then when those conditions are right, they sort of, come out of hiding, for lack of a better word, and then switch to the lytic cycle. So the lytic cycle is where it's just the death and destruction one, right? Meaning you're just going to have it go in, like, and a bacteriophage basically kills all and copies itself and then kills the cell on the way out. Whereas the lysogenic cycle would be like the covert secret spy who goes in and like infiltrates, and then later on, can take things out as needed. Other questions? Let's see, what else can I remind you of? Okay, looks like another question. If the virus eventually comes out of hiding and exits the cell, why doesn't it kill the cell at that point? I think it's just the idea that, well, when it does come out of hiding, like if it's switching from lysogenic to lytic, it it will very possibly kill the cell. But it just wouldn't necessarily immediately, because the way that the what you call it, sorry, I'm not making a lot of sense right now. It will kill the cell when it comes out of the lytic cycle. That's why we call it death and destruction. Yes, it can totally switch. I mean, some bacteriophages can switch, right? We call them temperate phages and virulent phages, but the idea that theoretically, yes, it could switch. It's not like a bacteriophage only does one or the other. But yeah, keep in mind those are in bacteriophage. which are the cells that infect bacteria, not the ones that infect us. Let's see, other stuff. Other questions? I want you to know about southern blotting in concept, but I mean, not all the itsy bitsy details. Hi. Yes, I can explain ecological succession. Basically, ecological succession is the idea that you're going to have these, over time, these very sort of predictable stages as something moves through. And so in the case of ecological succession, it's the idea that you might start off with like, well, there's primary and secondary succession, right? Primary is where you start off with like, there's not even soil, it's maybe just like bare rock. But over time, you develop all sorts of things. Okay, if mRNA binds which RNA, what is the protein form? I think they're talking about microRNAs, Tajin, or does no protein form? So if mRNA binds to RNA or microRNA binds to RNA, then yeah, you could definitely have a situation where you have no transcription, translation. Well, you probably would still have transcription because it's prior to that or after that, but translation could definitely be affected. So yes, in a lot of cases, I would imagine protein does not form. Sorry, contact lenses are kind of bothering me for a second. Let's see, what else? They're still finding out a lot about RNA. So I feel you probably hear more about that too in the coming years. Let me see, I'll go back to final quiz, final practice exam. Oh, wrong one. Definitely make sure you know your Punnett squares, that you can read a pedigree, that you can figure out stuff. Do you need to? I would just know that pH very generally what it is, right? Ion concentrations at lower pH means more acid. But I don't usually know too much about that one. Only whatever's in your notes, which is very, very low. Sorry. Allergies. Maybe a blink of it. Let's see. What else can I tell you? Or what other questions you have? Let's see. Sorry. Any other questions you guys have? Don't forget your semester 1 resources on the concept sheet, multiple choice. Go over your old exams, etc. Okay, can I go over why would you want to find, you would find a restriction site on DNA if you wanted to figure out, like, could it be cut up by a particular enzyme? But also it could be for mapping purposes, right? That you want to be able to draw a diagram of that particular plasmid or piece of DNA, and in doing so, you need to know where the restriction sites are. Can I please go over the difference between prokaryote and eukaryotic protein synthesis? I mean, process-wise, they're very similar, right? I mean, transcription comes in, or RNA polymerase comes in, and creates a piece of RNA. I would say a big difference in terms of the location, well, about protein synthesis is, first, you've got different promoters for prokaryotes versus eukaryotes. It's probably really the biggest one, to be honest. Yeah, I mean, in the case also of the prokaryote versus eukaryote, the eukaryote has a nucleus and the prokaryote doesn't, so you definitely wouldn't see things happening in the same locations. So I'm just going to try down questions. Looks like it's a pretty low question day, though. So anything? Anyone? Let's see though. Sorry, it's kind of loud. Other questions? Make sure you know about denaturing. So for nonpolar and polar amino acids, yeah, basically you're gonna look for the R group. You put the bonds within that R group. If it's only carbons and hydrogens and nothing else, it's a little simpler. But in other cases, you've got sort of different pieces. But in the end, it's about figuring out electronegativity and then figuring out what kind of bond it is. The sulfhydryl is the thiol, right? Technically, it's right on the cusp, I think, because if you look in the diagram, like the sulfur to hydrogen junction is right on the cusp. So for our sake, I was probably thinking we're very weakly polar. That's probably what I'd say. I guess I would say make sure you know your organelles, just in case. I don't know what else, guys. Sorry. Sorry, I'm afraid it's not the most chatty review. The properties of water include specific heat, surface area, adhesion, cohesion, and what else? So there's a lot of different properties of water. There's certainly the most basic one, which is that water is a universal solvent. Then you also have stuff like I said, yeah, cohesion, adhesion, which is like water sticks to itself, water sticks to other things. High specific heat, high heat vaporization. The idea of insulation in bodies of water, where you have like ice that floats to the top, it insulates everything else below it. And all of that is due to hydrogen bonding, right, between the atoms in water. I'm not sure what else you guys wanted to talk about. I would say for lab content, you want to know whatever's in your regular notes, right? So whatever you have in your regular notes about bacteria, about plasmids, about that kind of stuff. Yeah, there's no lab content. At least not the way there was in the first semester final, right? I mean, there's sort of a general understanding of the concepts that go into the labs, but I won't ask you specific lab-based questions. So no Alzheimer non-tables, no maps of plasmids and gels to read, things like that. What else? The phosphorus cycle. Yeah, I can, but honestly, I wouldn't worry too much about it. The big idea behind the phosphorus cycle, and really all the cycles, is the idea that like matter is energy flows when matter is recycled. So meaning like, weathering in rocks would like strip away, or not strip away, but like break down phosphorus, and it kind of gets taken into organisms, etc. So it just keeps going around and around. That's really the basics of what I need you to know. Especially since we didn't spend very long on it at all. Okay, what else? No problem. Anything else you guys have questions about? Do the splicism? I think they do. I mean, I have to assume it's a molecular signal, right? Because they're not just randomly cut up DNA, so something must tip them off. Yes. Gross and net prime productivity. Gross productivity is like they've just how much energy are you converting from sunlight, right? But then I think net prime productivity now it takes into account how much it costs you to do that. So just like to have a lemonade stand, you'd like, it costs you a little bit, like you have to sort of recoup that. But something you can also calculate, right? Or just give an example. What else? Which kind of DNA polymerase handles? I believe that would be DNA polymerase 2. Same idea. It's just a little faster. Which kind of DNA handles? Yes, I think I got that question. Okay, other excuse me, questions? I guess the thing I would just recommend you do is to yes, I can go over protein targeting, and then I will go over telomeres. So protein targeting is the idea that in order for a cell to leave the body, you have to mark it, right? That it doesn't know it's supposed to go anywhere. And so that's what happens here. Protein targeting is the idea that as soon as that gene starts to be transcribed, those proteins are getting ready to potentially go somewhere, right? So if you do transcription, but then translation, and now you have something that wants to leave, right? Then what you're going to need to do is, looks like, so basically you start making the protein, and then as it does, it's recognized by an SRP. The SRP drags that ribosome and protein over the rough ER, and now as it's made, it shoves it into the rough ER, etc. Okay, question from online says, when phosphotriester is placed in a high pH or high temp environment, it's no longer able to break down into substrates, which of the following bonds may be broken. Remember that typically with proteins and enzymes, you're going to be able to break secondary, tertiary, and quaternary structure, but it's a lot harder, not impossible, to break the first level of protein structure. Oh, I see, you're giving me the answers. So realistically, if we're talking about which of those bonds could be broken, but still have a working I'm trying to read the questions now. Let me see. Yeah, it's just talking about how you essentially messed up that enzyme. Yeah, I mean, the idea is like if you do disturb an area, certainly it might not come back. Things like but it could also, yes, lead to more diversity because there's more space for organisms before it didn't really have that foothold. They can now get their foot in the door. I don't get how to do that problem. Do you mean the one you just quoted up there? The thing is to think how often are those bonds broken during and so it's probably not going to be the peptide bond. It could be hydrogen bond. Disulfide ionic tend to be more well, disulfide is a covalent bond. In this case, they'll tell you it is the enzyme is placed in high pH and it can't do its job anymore. So that hopefully kind of narrows down sort of from a protein structural standpoint which one would be the best choice. Yeah, I mean, it could be hydrogen anionic. And I guess in this case, oh, hold on, let me look at the question. Sorry, I'm having trouble seeing today. Yeah, so essentially you could, pretty much the only one you probably wouldn't circle be peptide bond, just because, like I said, it's not typical that you change the primary structure. Okay, that's all the questions so far. What else? What else? Sorry. Apologize for rubbing my eyes. I should have taken my contacts out earlier. Sorry, guys. Let's see what else. I will look up at the practice exam again. See what else I can think of that you guys don't have in here yet. I think I talked about genetics. It's a pretty fast review of that. It seems pretty well covered. Well, let's see. We have 11 minutes left, but I don't know if anyone else has any questions. How hard would I say to find? I think it's hard just because it's a lot of material, right? It's not inherently meant to be hard, but I think it does become hard just because of the amount of information. Okay, difference between gel electrophoresis and LFRP. Technically, RFLP is what you actually see in your cells. It's not a procedure, whereas gel electrophoresis is, right? So gel electrophoresis is really just the idea that you have this gel matrix, and if you want to separate out the sizes of DNA, you're going to push it through. That's sort of gel electrophoresis. RFLP stands for restriction fragment length polymorphism. That's really describing something you're going to have in your cells. It's not really something you would sort of do out of context, though. And it's the idea that because your DNA is different from anyone else's, it'll be cut up by restriction enzymes differently. When there is a promoter sequence that's 25, yeah, you go 25, and then you go like 25, 24, 22, 22, 21, 2019 is all the way down, yes, to -1 +1. I hope that answers your question. Kind of all over the place today. You do not need to memorize the promoter sequences. If you need those, I will give those to you. Let's see. I'm not sure what else you guys have questions about. Wait, like 9 more minutes. So, any other questions? Let's see. Let me see what's going on the website. I guess there's a key. There's a concept sheet. Let me see. What else can I talk to you guys about? Let's see. In what conditions would a cell have to? So think of it as not all cells are actually dividing all the time. They would sort of just potentially be in like a holding pattern, which is kind of what I think of G0 is. So probably a good number of cells could be in G0. So yeah, hopefully that helps. There's no specific condition when you'd have to, but you might enter. We're looking at like 6 more minutes. Any questions? Okay. Any other questions? It's very similar. It's just shorter. A lot shorter, like half as short. And then the second half of your final number will be on the 21st of May, which is a Tuesday. You're good. Oh, so it's a lot shorter, right? It's like 45 questions, whereas your other final was like 90 questions. So in that sense, it should be very doable for you to do in 45 minutes. Let's see. Other questions? Something else that I'm still trying to work out is our AP exam calculator information, but for now, just a reminder, make sure you have your let's see, I don't know what I was saying. Sorry. Any last questions? Like 5 minutes left. I guess you could say then the first half of your final is worth 12.5%, and the second half is worth 12.5%, also the combined total 25%. Oh yes, endocytosis and exocytosis, sure. So they are both forms of cell transport, right? They're sort of both involve vesicles. With endocytosis, it's the idea that sort of like there's a molecule to be eaten, the membrane kind of pinches in on itself, becomes a vesicle contacts with the compounds. So basically, like it's the vesicle contains the compounds, and now they're spit out into the cell. Exocytosis is sort of the opposite, right? Where you have some sort of molecule you want to spit out, and then that is in vesicles, and then fuses with the membrane, it gets expelled. Yes, cholesterol and temperature and level of fluidity. So essentially, remember, in order for a membrane to work, it has to be fluid, and that means things have to be moved around. So it's a big part of it. But cholesterol sort of balances that out, makes it more solid at room temperature. And if it's more solid, obviously, it's less. Fluidity levels are down. Anything else? No problem. Okay. Oh, lots of questions. Okay. No, don't worry about the pipettes and lab equipment. That's like more lab specific. How do temperatures affect PMS? Basically, the PCR you have in the DNA polymerase you have in polymerase in PCR is a heat stable PCR. So heat stable enzyme. So that's why it doesn't become an issue that you have at those high temperatures. The way a karyotype works is basically you want to count the chromosomes. So you take a picture of the chromosomes in M phase, mitosis, and then you'll start to see differences where it might somebody or such. Oh, sorry. I was talking about karyotypes today. I got distracted. So karyotypes basically is like you find cells, I think they're in like M phase, you take a picture of the chromosomes, and then you cut the chromosomes and you match them up to make sure everything's there. That's when you might see an extra or something's missing. Okay, any remaining questions? Sorry it's too late again. You're welcome. Okay, so it's not really looking like people have too many questions, so I'll probably wrap it up soon. It's pretty late. You all should go to sleep. I should probably too. If you have any questions, I know. I'm assuming people will watch it later. I hope. Otherwise, we'll rethink this. I don't know. Anyways, you guys should go to sleep. Get some rest. And I will talk to you guys later. And if you have any questions, you can ask me in person or email me. Yeah, hopefully they're not stressing, huh? No, I hope not crying either. But I hope you all have a good night, okay? And I will talk to you guys soon. Yeah, 7th period, I'll see you tomorrow. Yes, it's okay. You can still study. You'll be okay. All right? All right, guys, I will see you tomorrow. Bye-bye. Good night.